I appreciate the invitation to come here. John Green said that I needed to come down and talk to you all about a rather interesting aspect of cancer that we're quite interested in Moffitt. You might ask why thoracic surgeons involved with microorganisms, since most thoracic surgeons couldn't really spell HIV, but nonetheless, I began to realize after a lot of reading that even some of us old guys can learn things. In January, we founded the Center for Infection Research at Moffitt. There's an enormous number of people involved in infection research in terms of the etiology of cancer. And Anna Juliana is one of the world's experts. She's the director. And John Green and I are co-directors of this. And I won't say much more about that except that we're in the process of a lot of different studies looking at cancers. I chose, obviously, lung cancer. And lung cancer probably is one of the very prevalent cancers that has really very little information about whether or not it's involved, or its etiology in some respects involves microorganisms. And let me just talk a little bit about microorganisms and disease. I'll beat you over the head just a moment about the numbers, because I think a lot of people don't realize it. But lung cancer, again, is by far the biggest killer in the United States, actually in the world. About 161,000 people die of lung cancer every year. That equals the next 4 cancers combined. They don't have as good a PR firm, so people are focused on a lot of other cancers, but lung cancers are still our main killer. And the other thing to realize is that you don't have to be a smoker to get lung cancer. You just talk to the 58-year-old woman I saw this afternoon, lifelong non-smoker, healthy, developed a cough, and has got stage 3B lung cancer diagnosed from a little needle biopsy right up here this afternoon. So she's devastated by the disease. And females particularly are the ones at risk for the ones who are non-smokers who get lung cancer. This is the standard etiology of lung cancer, what is considered to be the party line. Currently around 80 to 90% of current or former smokers get lung cancer. We've said that probably some of this is secondhand smoke, which is probably realistic. A family history increases your risk. Genetic susceptibility probably comes from polymorphisms. There's a lot of those, particularly in females, that may have them have a higher risk of getting lung cancer. That's a lot of conjecture, and there's some changes. Radiation exposure, radon, gamma radiation, a very rare cause of lung cancer, but probably some radon might, but I wouldn't count on, unless you have a good trial lawyer can maybe show you, but that's about the only ones who can really prove that. Advancing age, there's declining immune system, and lung cancer increases with declining age, as do most cancers. There's a number of environmental carcinogens that might cause lung cancer or contribute to it. Asbestos probably doesn't really cause lung cancer. It just increases your risk considerably, about 10 times if you're a smoker or a former smoker. Chronic inflammation, and actually chronic inflammation and recurrent infections appear to be the pathway that we're beginning to realize with most cancers. In fact, as far as recurrent infections, if you have a number of pneumonias, non-smoker, it increases your risk of getting lung cancer by itself. Chronic obstructive pulmonary disease, which is usually involved with chronic inflammation, and most people predispose you to lung cancer. And all of this, we think, leads to a pathway of activation of oncogenes or inactivation of tumor suppressor genes. And this is the party line, and probably fairly accurate. But what about an infectious cause? And that's where we started looking at this. And the reason we're looking at this is because there's a number of other cancers that have been shown to be caused by various microorganisms, and I'll mention that in just a moment. But what about an infection cause? Isaac Newton was probably the one who gave us the most realistic view that no great discovery was ever made without a bold guess. So we have to make a bold guess and not just go with the flow. Because the party line is it has nothing to do with microorganisms, and I think a lot of us are beginning to realize that it may be different. Let me talk just briefly. Surgeons are always focused on history, at least the history of medicine, so I'm going to say a couple things about history of infectious disease and discovery of infectious diseases. Hippocrates, the old father of medicine, Western medicine, was one who really proposed that and correctly believed diseases were caused naturally. And before, at that time, it was felt it was by the gods or superstition. But he felt all of these diseases, which are mostly infectious diseases, were caused by the imbalance of the four humors: yellow bile, black bile, phlegm, and blood. Nice while you're eating here. I appreciate that. Actually, a couple hundred years earlier than that, however, in India, the Indian sage Mahavira described what he called Nagoda, which was a realm where all the lowest life forms, submicroscopic creatures, lived. I think I've stayed at the Hotel Nagoda before this past summer. He also taught that creatures, get this back, that creatures permeate every place in the universe. In other words, he was talking about microorganisms, and this was 2,500 years ago. It was actually another 1,400 years, 1,500 years, until Avicenna, this is the really renowned Persian physician, described contagious diseases in his textbook, The Canon of Medicine. Just to show you how books don't get out of date, this particular textbook was used for the next 200 years in teaching medical training. And he advocated the quarantine to limit the spread of infectious diseases. Also, he was a big proponent of exercise as a way to help keep yourself healthy. This was quite a number of years ago, long before it became popular now. It was not until van Leeuwenhoek actually demonstrated or showed the single-cell organisms in the 17th century, actually showed them on his own microscope. He described them, calling them animalcules, bacteria, infusoria, muscle fibers, and even sperm. He was able to show, which caused quite a stir among the religious leaders at that time. But he was able to demonstrate this, and others did the same thing using his techniques, so he was vindicated. It was really Louis Pasteur in the 19th century that really laid the foundation of the germ theory of disease, developed a technique, for example, to heat milk to kill microorganisms, which we now call pasteurization, and he created the first vaccines for rabies and anthrax. However, the most well-known of the 19th century physicians is Robert Koch, and you've all learned this all to begin with, Koch's postulates. And Robert Koch isolated the anthrax bacillus, TB bacillus, and cholera, showed that these caused the various diseases. And he came up with what's called his postulates in the late 19th century, 1893. And these are criteria he published that he felt must be fulfilled in order to establish the microorganism caused disease. And these are the postulates. First of all, you had to be able to isolate the microorganism in all cases of disease and be absent in healthy animals or healthy people. The microorganism you had to be able to put in a pure culture outside of the animal. Then his third postulate was that microorganisms in this culture could be reintroduced back into the animal to cause the infection and create the original disease. And then finally, the editors probably of the journal, this is not probably his postulate, but the editors also added number 4, and that you have to be able to take the organisms out again from the inoculated animal and culture them again. Well, this was all very well and good when he published these in 1893. Soon after it, however, he realized himself that his postulates were fallible. The first postulate, that everyone who had the organism has disease, he realized there was a lot of healthy carriers of cholera, and subsequently a lot more different asymptomatic carriers have been found in viruses, polio, hep C, etc. Then there's a number of these organisms can't be grown in cultures. You're well known. Prions, such as those cause Creutzfeldt-Jakob disease, Mycobacterium leprae. It can be grown in the footpad of an armadillo, but it's very difficult to grow. And there's other organisms that can't be grown in culture. So satisfying Koch's postulates is sort of theoretically preferable, but not necessary to show the causation of diseases. There's a lot of limitations, and I'll just briefly mention some of these. If you look at it very carefully, you can't grow a lot of them in culture. There's a limited host range, so you can't actually put some of these organisms in animals, so you can't do that. There's numerous carrier states, a lot of carrier states. Some microbes can cause distant injury by releasing a toxin such as tetanus, and via some immune mechanisms after the microbe has already disappeared. Microbes can switch off and on some disease-causing genes. They can require a coexistent infection, such like hepatitis D. You can't have until you actually have another microorganism, hepatitis B. there's certain microbes that don't cause disease in all the infected organisms, such as HIV, EB virus. 90% of the people, adults in the United States, are felt to have either have EB virus in them or evidence that they had been exposed to it, yet they don't have disease. And probably much of the Hodgkin's disease is caused by EB virus. There's a lot of latent infection states. The most typical one is herpes zoster. The other thing, the problem is the microbes are so ubiquitous inside the body, just finding the microbe there doesn't necessarily mean it causes disease. Is it a commensal or a pathologic organism? Now, techniques of molecular biology are now available that we can search for evidence of microbial DNA that was hiding in the genome. But then there's a lot of problems there. The extreme sensitivity of PCR. PCR amplifies DNA approximately 1 billion times in 2 hours, so you can have only a very tiny amount, and yet you can find the DNA. What does that mean? There's clinically insignificant organisms can be found all the time, so trying to, again, go back to this idea of causality is a difficult problem. There's a lot of irrelevant contaminants, and then a lot of times the DNA sequences are found different from the anatomic location of the disease, and what does that mean? And there's a lot of disease models for some of these, and particularly when we're talking about cancer. So the question is, in the absence of pure amounts of these microorganisms or the ability to grow them in culture, can just DNA sequencing provide convincing evidence of causality? Well, in late 1990s, Relman came up with some ideas of revising the Koch's postulates to be really what's considered molecular Koch's postulates, to try to be able to prove causation of disease from molecular evidence. And these postulates are that a sequence of nucleic acids belonging to the pathogen should be present. Again, all of these should be present in most cases. There's no absolutes in this, unlike in Koch's postulates. Few or no copies of the nucleic acid sequences should be found in normal individuals or in tissues that didn't have any disease. The copy number of these sequences should decrease or disappear when you resolve the disease. The number of copies of the nucleic acid sequences should correlate with the severity. And the nature of the pathogen that was suggested by these nucleic acids sequences should be characteristic of the microbe family that you're coming up with. You should have an effort to visualize the pathogen-associated sequences directly. And again, talking about in situ hybridization. And finally, the sequences, sequence-based forms of evidence you should be able to reproduce in other laboratories, which is the key, and sometimes that's been very difficult to happen. This is Zur Hausen. He won the Nobel Prize in Medicine in 2008 for his discovery of human papillomaviruses causing cervical cancer. He's the chair of the Scientific Advisory Committee for the Center for Infection Research in Moffitt. In fact, he's going to be over here in about a month for their first visit. Dr. Zur Hausen has an extraordinary book, Infections Causing Human Cancers. It came out in 2011. An extraordinary book intents of the documentation of all of the data really known about this entire field. And in his book, the first part of it, he really talks about what he considers, after reviewing all of this literature, the criteria for a causal role in direct carcinogenesis by microorganisms. He feels that you have to have the regular presence of the genome or parts of the infectious agents in every cancer cell that you find to really be able to prove this is the case. Transfection of the nucleic acid in tissue cultures or lab animals should result in cell immortalization or tumor induction. And some of these are, again, quite difficult to do. Excision of the nucleic acid from the cells should lead to reversal of the immortalized cell and the malignant phenotype. And finally, you should be able to use some epidemiological case controls and prospective studies to identify this agent as a major risk factor for the respective tumor type. Now that's for direct carcinogenesis. The problem is indirect carcinogens, and that's another problem, the hit and run mechanisms. HIV causes immunosuppression, which induces immunosuppression and then allows perhaps another virus such as EB virus or HHV8 to induce malignant transformation. Been shown very well. It can prevent apoptosis by viruses, such as the cutaneous papilloma infections that could result in squamous cell carcinoma of the skin. Chronic inflammation, increased radicals, carcinogen metabolites, and other factors by these various microorganisms may be prime drivers of developing cancer in these tissues. So the microorganism indirectly causes the cancer. Mutations, chromosomal rearrangements of the host DNA that are induced by the infections could result in cancer. Again, an indirect cause. And then persisting viral infections can introduce a long-lasting chromosomal instability. And this is how probably some of these viruses actually work. So these are some of the different ways that infectious agents can act as carcinogens. Based on his study, and this is actually taken from his book, or I created from his book, the current number of infections that are, or tumors that are presently linked to infections, about 20% of all cancer is felt to be infectious in origin or induced by infectious agents. The prime ones, of course, are HPV, cancer of the cervix, perianal, anal cancers, vaginal cancers, vulvar, penile cancers, head and neck cancers. Head and neck cancer is one of the most rapidly rising cancers, and at least 78% of these cancers are caused by HPV. And it's dramatically going up. And they talk to the head and neck people, and they're seeing an awful lot of young people who have HPV-induced cancers. So it's quite a tremendous rise. Of course, hepatitis B and C can cause hepatocellular carcinomas, cholangiocarcinomas from some of the Clonorchis. So there's a number of different cancers. I should draw your attention, because I'll mention this in just a minute. It's felt that seminomas may be related to endogenous retroviruses, and I'll talk about that in just a few minutes when we talk about some of the preliminary studies we've found with lung cancer. So what clinical evidence makes you think? What's made me think? As a thoracic surgeon, I've been doing this for almost 4 decades. What makes me think that lung cancer could be caused by microorganisms? I'm just going to show you a few things that sure don't seem to be, they seem to be something that they would expect to come from a microorganism. For example, 10 to 15% of the victims, lifelong non-smokers, no predisposing factors, healthy, lady I saw today, BMI 21, 58 years old, no cancer in her family, no nothing. She couldn't leave a more pristine life. Is on an exercise program and comes in and has bronchoalveolar cancer, filling much of her upper lobe, a mediastinum full of lymph nodes and supraclavicular lymph nodes, and just out of the blue. Why did she get that? The multicentric pattern of some of the cancers is very suspicious, particularly bronchoalveolar cancer. That's the one cancer that I'm sure is going to turn out to be a microorganism-induced cancer. These same cancers can give you this very interesting diffuse mnemonic pattern of cancer. They have lipidic spread of the bronchoalveolar carcinoma, for example, spreads directly in the airways along intact alveolar walls, not through the lymphatics, not through the blood. It spreads just like a bacterial organism would do, or a viral organism would do, erogenously. Head and neck cancer patients commonly develop subsequent primary lung cancers. It's always the cell type, same cell type, squamous cell, head and neck. 25% of them will develop a lung cancer. Let me show you a few pictures. Everyone likes to see chest x-rays and CAT scans. This is a typical lung cancer. This is a 82 or 83-year-old heavy smoker, cough, dyspnea, emphysema, and he has this mass in his lung. He had a right middle lobectomy. It was stage 1 squamous cell carcinoma. Classic cancer of the lung. 5-year cure rate for this is about 85%. Here's another one. A little harder to see in the light. 69-year-old former smoker, prior head and neck squamous cell cancer, had a cough and hemoptysis, and he had this giant cancer right here at the distal left mainstem bronchus, and he had a left pneumonectomy. He was also invading the pericardium, took out his pericardiectomy with it, and he had a 7.5 centimeter large cell undifferentiated cancer. It was large cell, that's right, and invading the pericardium. His cure rate's about 50%, and he's done well with it. This is what you think of as lung cancer. How about this lady? 55-year-old lifelong non-smoker, had a little mild cough, found to have this kind of a hazy area here. I used to think of this, looks like it kind of had bubbles in the middle of it because it's not a very solid tumor. This was a bronchoalveolar carcinoma, 2.5 centimeters in diameter. Interesting enough, she had some hilar metastasis. This is a dietitian at Moffitt. We operated on about 5 years ago, and she's currently free of any disease and hopefully will continue to do so. This one, how about this? What do you think this is? This looks like somebody came in that has a Klebsiella pneumonia, or could this be a patient who had a bronchoalveolar carcinoma with pneumonic spread, tumor in the airways? And that's what it turns out to be. We took out her lobe of her lung. You can see the major fissure between the middle and lower lobes right there. It's only mildly PET scan positive. You can barely sort of make out on this a little bit of glucose uptake. These bronchoalveolar cancers commonly will not have a lot of glucose uptake on a PET scan. In fact, a lot of them will be completely negative and there's a cancer. How about this lady? This is a lady I operated on in 2008. She's a lady from Tallahassee. She had this lesion, bronchoalveolar carcinoma. Then she had this little hazy area in the upper lobe. Then on the left upper lobe, on the opposite side, she had this one and another one. These were fairly stable, hadn't really changed much, and this was getting bigger. We went ahead and did a right lower lobectomy and wedged out what was in the upper lobe. Both were bronchoalveolar carcinomas. One was 8.5 centimeters, the other was 1.9 centimeters. She was treated with some adjuvant chemotherapy, and it was felt these were likely cancers as well. She got some adjuvant chemotherapy, hummed along, and had a little headache once. About 2 years later, had this brain met, which was resected. She's still running around. I see her at Moffitt all the time. She's still doing well. Bronchoalveolar carcinoma can have a relatively indolent course that they are actually, even with stage 4 disease, as this lady had when she actually presented, can still do all right. But it's a very interesting disease. So some of these presentations make me think more in terms of could they be actually caused, and particularly the pneumonic spread. That's the one that really got my attention. It just seemed like a pneumonia. So the hypothesis we might have regarding lung cancer causation in microorganisms, well, epidemiologic research has really provided some evidence that tissue damage, again, caused by chronic inflammation, can initiate or promote development of cancer. The lung is the site where you have repeated inflammatory insults. I just got over bronchitis, probably had a little pneumonitis at the same time. We get viruses, bacteria, and then, of course, tobacco smoke and all the other irritants in the lungs. Reactive species or nitrogen species that produced by activated neutrophils happens all the time. They bind to DNA, can cause genomic alterations, and can lead to transformation into a cancer. Inflammation can be conceived as a cancer initiator promoter, and this again is the big field now in oncology. Most feel that inflammation is the primary pathway to cancer. Tissue repair is associated with a lot of cellular proliferation, pro-inflammatory cytokines, all sorts of changes and upregulations of various genes that can lead the same kind of thing you see with cancer. And then, of course, angiogenesis, a common feature needed for tissue repair, is also very crucial for tumor growth. What about some of the studies that have been out there as far as lung cancer is concerned? These are all molecular studies, just to show you how it kind of jumps around the whole place. In China, 2008, they looked at 313 fresh tumors. They found HPV 16 and 18, looking to see is HPV, which causes cervical cancer and head and neck cancer, is it present in lung cancer? And this has really been the only major virus that's been looked at. And they found over half of the squamous cell cancers and 25% of the adenos, they could find genomic evidence, at least nucleic acid evidence of HPV 16 or 18, which are the two of the bad guys in HPV. And then at the NIH, 3 years later, they did 450 formalin-fixed paraffin-embedded lung tumors and found no evidence, zero, at 450. And there they looked at the gene sequences E6 and E7 by RT-PCR. 2007 in Italy, using some other molecular techniques, they only looked at 6 tumors, but they found, again, this is all that's in the literature, 83% of them had the E6 and E7 oncogenes of HPV that were demonstrated by looking at the RNA transcripts. Well, then they looked at the reviews. The review now taking all of the studies, and 86 studies, they were done a variety of different techniques. There were 2,400 over 24-hour lung cancers. And it went anywhere from 0% to 100% had HPV detection rate, averaging about 21%. And most recently, again back at the NIH, they looked at 290 lung cancer cases and found little evidence of some other viruses they're looking for. And microRNAs, DNA using microarrays, and quantitative PCR. Now, why do we have such variability? Well, a lot of factors could account for this. There's a lot of genetic variations. There's some environmental exposures. There's a lot of different methodological approaches, inappropriate primers. The problem with it, PCR, obviously, you have to choose the primers exactly correct, or you'll miss what you're trying to find. So there's a number of technical problems that might make this so difficult to see. We had the opportunity to be involved with the Lawrence Livermore Laboratory. I do find there's something missing there. It's a pan-microbial array. That's all right. I actually contacted these folks, and the Lawrence Livermore National Laboratory, you may recognize that was involved in creating the atom bomb a number of years ago. They do not create bombs now. Fortunately, they're involved in a lot of other things, a lot better for us. And they've created probably the most sensitive pan-microbial detection array in the country, probably the world. And their microarray is actually now the next generation of it has 388,000 probes that probe at least 50 probes sequence of all of the known viral families, all of the known bacterial families. So they can take nucleic acid, put it on this little array. They let it sit and percolate for 17 hours, and then the computer tells you what's positive. So it's remarkable. It's almost seemed like Harry Potter magic, but it's been validated and is extraordinary because they validate it with PCR. They say what's on here, and then they use PCR, and the concordance rate's 100%. So they say it's on the array, PCR verifies it 100%. They've done a number of studies. They have not done any studies with cancer whatsoever. That's why they were very excited to get involved with this. What they've done, for example, they've taken some of the known vaccines and looked to see what was in the vaccine. Was it what you said it was? And actually, there's a rotavirus vaccine they published about a year ago, had a bunch of pig virus in it, and which appears to be what they call a harmless pig virus. I don't remember the name of the virus, but it was called harmless. I'm sure that's what the government told us, at least. So we designed a study, which is actually going on right now, and I want to explain a little bit about what we decided to do. First of all, we need to find out which are the suspect microorganisms, because they may not be HPV, maybe something completely different. And so this allows us to really screen and look who are the suspects. So what we've done, the Moffitt has an enormous, in fact, I think it's the largest bank of frozen tumors in the world. And we've taken, from the Moffitt Tumor Bank, we're taking 10 squamous cell carcinomas, 10 adenocarcinomas, 10 bronchoalveolar carcinomas, and then we have a series of cancers where we've taken out the primary, and we've also taken out an isolated solitary metastasis. And we do that occasionally, and it's mostly a brain metastasis. And we have matched lung cancer and brain metastasis. So we have 10 of those where we're going to look at the primary and then look at the corresponding brain metastasis, looking for microorganisms. And then we're going to also look at lifelong non-smokers. And then we have we're going to use 10 normal lung tissue specimens for control, run it through the microarray. We're going to screen for microorganisms, looking at the DNA and RNA using the Lawrence Laboratory pan-microbial detection array. And we've actually started this just so you know sort of what the data looks like. This is from one squamous cell carcinoma. And I won't go through all of how this actually works, and actually it takes a good biostatistician to sort of explain things. The ones in red are the hits that have a high incidence and a high probability, a 99%, or actually here 95% probability that there are nucleic acid sequences from these various viruses. Several interesting ones which have a high incidence so far we've only done 10 squamous cell cancers. We just sent out some adenos. Human T lymphotrophic virus number 2, HTLV2. HTLV1, of course, causes some leukemias. This is HTLV2. It's found in a lot of these squamous cells. The human endogenous retroviruses is found in all of the people. Now the question is, are those just innocent bystanders and and I'm going to mention this second about endogenous retroviruses, those who may not know much about it, I recently learned. Interesting enough, hepatitis B is found in most of these specimens as well. And the other thing that's kind of interesting, HPV 57 is found in a high instance of these same specimens. In fact, the first 10 squamous cells, 7 of the 10 had HTLV2, 6 of the 10 had human papillomavirus type 57. All of them had hepatitis B virus, and at least 8 of the 10 they found evidence of the hepatitis delta virus. And then the other one is the human endogenous retroviruses, 3 of them. Now, the people out of Lawrence Livermore Laboratory said they think these are most likely just background. And the reason they even put probes for this on their microarray is because they're not always looking at human specimens, but they assume that those are going to be probably positive in most tissue. So we're interested to see what our normal lung tissue, whether they're actually going to show up in all the normal lung tissue, and probably can ignore them if that's the case. Interesting enough, they found no bacteria whatsoever in the tumor, so which is good in terms of contamination in processing. The data is very intriguing, but we're just waiting for some of the more tumor samples to come up. By the way, the human endogenous retroviruses, these are a family of viruses which are very similar to the exogenous retroviruses that you're quite well aware of, and it's felt that these joined the genome in most people eons ago and have been passed down evolutionarily and have perhaps some biologic benefits, may confirm some benefits, no one's really sure. On the other hand, there are some indications that it might actually cause disease, and like I mentioned, seminoma is one of the diseases that may be caused by, or at least in some seminomas, might be caused by this particular virus. So we'll have to wait and see if this is going to have any bearing on lung cancer. After we identify what are the target microorganisms, then we plan on, from the microarray, then Dr. Scott Antonia's lab, who's collaborators with us, co-investigators, are going to design appropriate primers and do a confirmatory PCR just to verify that what they found in the microarray is what's actually found. And then after the microorganism's presence is confirmed by PCR, then they're going to design a Western blot analysis and see if there's any viral protein expression from the suspect organisms. Now that would be a lot more of an indication that the microorganism is active, because again, just because you find a piece of nucleic acid from virus X, is it actually doing anything? Was it an innocent bystander? Did it get sort of mixed up in that? I think probably the thing to realize, of course, is that the lung is a contaminated organ. So if you didn't find virus in our tumor specimen, there's something wrong with your technique, because it will be contaminated. It will have organisms in it. However, theoretically, the brain is sterile. So this is the importance of looking at brain metastasis, because if we find microorganisms in the brain metastasis, particularly if they're the same microorganisms that's in the matched lung cancer, then that gives very exciting potential that there may be something involved. First of all, the microorganism may be a true pathogen, and secondly, it may be involved in the process of metastasis. So we'll wait and see. This is ongoing, and one of the things, if you ever try to arrange for collaboration with a government laboratory, you'll find it's probably the worst thing in the world. It was an extraordinary thing. We just put in a grant, and the overhead from the Lawrence Livermore National Laboratory is 105%. Now, for those who don't know much about grants, when you put in a grant for so much, say $250,000, then Moffitt says, well, we have to also pay for the lights and taking care of everything, so this is the overhead. So the overhead for most grants is about 45%. So if you get a $250,000 grant from the NIH, you also get another 45% added on to that for overhead. The Lawrence Livermore National Laboratory, which is the standard government overhead, is 105%, which just shows you they can do it better and a lot more expensive. Yeah. Finally, we're going to look at all the clinical information from these patients, which it hasn't really been done, to see if there's any evidence, any correlations with histology, prior cancers, and any long-term treatment results, because we know all these patients are. And finally, we have an interesting thing. When Merck got together with Moffitt to create the Total Cancer Care Initiative, they went ahead and did microarrays or I should say they did gene expression profile data on an enormous number of cancers, of which 2,000 were lung cancers. And we're choosing the cancers we're using of those who have gene expression profiles. And we're going to look at those after we're done with this to try to figure out if you can find any particular gene expression profiles that might have any correlation with what we find in microorganisms. And this is of course, has never been done as well. By the way, just to do a gene expression profile on one cancer costs about $1,000, and they've done probably just in lung cancer probably at least $2 million worth. So, and there's no data from that yet that's been useful, but it's just starting to get evaluated. So the question comes back again to my first key question. In the absence of any pure amounts of microorganisms or ability to grow these in laboratory culture, can DNA sequencing methods, which we're using, provide any convincing evidence of causality? And that's the big key issue. And the answer is it doesn't, but we have to start someplace, and we're trying to find who the organisms are. And my favorite quotation, one of my favorite writers, you've probably seen this before, you saw it in the movies, when you've eliminated the impossible, whatever remains, however improbable, must be the truth. Sherlock Holmes.